So we were discussing about the chromosomes. We have already discussed about the mitosis. Today we have to start with the meiosis. Okay, there are two types of cell division, that is mitosis and meiosis. We have already discussed about the mitosis and its significance also. We have seen the difference of mitosis in plant cell and animal cell. Today we are going to start with the another type of cell division, and that type of cell division is known as meiosis. So meiosis is a special type of cell division which occurs in sexually reproducing organisms. Why it is telling that sexually reproducing organisms? Because this type of cell division will occur in the reproductive cells or the germinal cells. Okay, and the germinal cells are also known as gametes. So why we call it as gametes? Gametes are the reproductive cells which has half of the number of the chromosome as compared to the somatic cells. Okay, so gametes, so which of these cells will be gametes? That is a sperm cell and the egg cell or the ovum cell are the two types of gamete. A sperm cell is the male gamete and egg cell is the female gamete. So this type of cell division will occur in the germinal cells and helps in the formation of gametes. Gametes will have the haploid set of chromosomes. It involves 2 rounds of division that ultimately results in 4 cells with only one of each chromosome. Okay, so there will be meiosis 1 and meiosis 2, and from one parent cell we will get 4 daughter cells, and these 4 daughter cells will have half set of chromosome as compared to the parent cell. Okay, now we must have to understand what is homologous chromosome, because on this concept only the crossing over phenomena can be understood. So homologous chromosomes are also known as homologs, which consist of the members of any given chromosome pair. So one member of the homolog is the chromosome from the mother cell and the other member is from the father cell. So during meiosis, homologs will pair together. Like this is the homologous pair of chromosome. For example, red one is the maternal chromosome and blue one is the paternal chromosome. So these two are the homologous chromosomes. These red ones are the sister chromatids. Blue ones are the sister chromatids. But red and blue will be known as non-sister chromatid. So the crossing over will occur in the non-sister chromatids of the homologous pair of chromosomes. So one chromosome of each homologous pair comes from the mother called maternal chromosome, and one from the father is known as paternal chromosome. Okay, like here, one, for example, red one is the maternal chromosome and blue one is the paternal chromosome. Okay, homologous chromosomes are similar but not identical. Okay, like example, homologous chromosome number, for example, this is 1 and this is also chromosome number 1. This is coming from father side, this is coming from mother side. So they are identical to each other. It means they will have the same type of locus on which the genes are present, but the alleles for each trait will not be the same. So each carries the same gene in the same order. Like here is the gene for the eye color and here is the gene for the hair color. Okay, but the alleles may be different. Maybe this is for black eye and this is for brown eye. Okay, so the two alternative forms of one phenotypical character is known as alleles. Here for a hair color, this allele may be for black hair, black hair color, and this may be for the burgundy hair color. So it has the same locus but the alleles will be different. Okay, so each carries the same gene in the same order but the alleles for each trait may be same or may not be same. They may be same also and they may not be same. Okay, so these two are the homologous chromosomes. This is the centromere, this is the kinetochore. These two red ones are the sister chromatid, but the red one and the blue one is the non-sister chromatid. Here homologous chromosome is shown. So homologous chromosome consists of 4 chromatids, 1 chromatid, 2 chromatid, 3 chromatid, 4 chromatid. So that is why it is known as tetrad. Okay. One chromosome comes from the paternal side, one chromosome comes from the maternal side. They will have the same order of genes, but the alleles may be different. What is the significance of meiosis? Meiosis is the reductional cell division which solves the problem of polyploidy. What is the problem of polyploidy? For example, in case of humans, if the number of chromosome in humans is 46, if a sperm is also bringing 46 chromosome and the ovum is also bringing 46 chromosome, then the total number of chromosome in the zygote will be 92. Then will it be the Homo sapiens? We know that species, the characteristic feature of species is what? They have a fixed set of chromosomes or fixed number of chromosomes. So for being Homo sapiens, how many chromosomes should be there? 46. So that is why so if there will be no meiosis, it will increase after each and every generation. It will multiply itself. 46 into 2, 92. Polyploidy, then again 92 into 2, that is 184. So that is, such a condition is known as polyploidy. But what happens due to meiosis? The number of chromosome in the gametes is reduced to half. Okay, so that is why the meiosis is the reductional cell division, and when the number of chromosomes is reduced to half in the gametes, that is the male cell and the female cell, that is the sperm and the ovum. So when they fuse together during the process of fertilization, 23 plus 23, it will come to 46. So the number of chromosome is maintained generation after generation through the meiosis which occurs in the gametes or the reproductive cells. Now, this type of cell division is bringing genetic diversity. Diversity, it means change. So, genetic diversity is created by two phenomena, that is crossing over and independent assortment of chromosomes. So let's see about crossing over. So crossing over will occur in prophase 1 of the meiosis. As I said, meiosis, in case of meiosis, the karyokinesis occurs 2 times. So that will be meiosis 1 and meiosis 2. So in meiosis 1, the first prophase, in that stage crossing over will occur. Then it is defined as the exchange of chromosome segments between non-sister chromatids. It should be non. It is written here no. Non-sister chromatids in meiosis. So this is the one. For example, this is paternal and this is maternal. Such a chromosome pair is the homologous chromosome pair. So these two will be the sister chromatid, but this one and this one will be the non-sister chromatid. So crossing over will occur in the non-sister chromatids of the homologous chromosomes, and it will this X-shaped structure formed during the crossing over is known as chiasmata. The pairing of two chromosomes together creates a tetrad or bivalent. So 4 chromatids are there, 1, 2, 3, 4. So that is why it is known as tetrad or bivalent. Bi, it means 2. So they have the 2 copies of the similar type of chromosome. Okay. Then the point where the chromosome attached is known as chiasmata. Okay. So this X-shaped structure formed during the crossing over is known as chiasmata. Chiasmata occurs, or chiasmata is formed in the non-sister chromatids of homologous chromosomes. So after the exchange of genes, new combination is formed. Okay. Understood it? The point where the chromosome is attached is called the chiasmata. The end result is the two recombinant chromosomes which have a different combination of gene than their parents. So in the parents, it had capital A, capital B, but here in the recombinant chromatids, it is capital A and small b and small b and small a and capital B. Whereas these two are the non-recombinant chromatids and the inner ones are the recombinant chromatids. So this is how the genetic variation is brought, and variation is the raw material for evolution, and evolution is essential for the better adjustment or adaptation of any species in the changing environmental conditions. Now we have to see independent assortment. So homologous chromosomes will line up randomly in the metaphase 1. So the chromosomes that end up in each daughter cells after separation will vary. So red one is denoting the maternal set of chromosome, blue one is denoting the paternal set of chromosome. So this is the possibility 1 and this is the possibility 2. So blue blue and red red. Here red blue and red blue. So in this combination 1, after the result, this may be the combination 2, this may be the combination 3, or this may be the combination 4. Out of any combination, any of type of combination can be formed. So this is known as independent assortment. Now we are coming to the last part of the topic, that is the comparison between the mitosis and meiosis. Now what is common? Both mitosis and meiosis will create new cells and will follow the same steps, that is karyokinesis and then cytokinesis, and both will start with one parent cell. Okay, but what is the difference? Mitosis occurs in all organisms except viruses, whereas meiosis will be found in animals and plants and fungi, where the sexual reproduction will occur. Then mitosis creates the body cells, whereas the meiosis will produce the sex cells. Right now we have seen. Then there is only one karyokinesis and cytokinesis, whereas here there is two karyokinesis and one cytokinesis. No recombination or crossing over occurs in mitosis, whereas recombination or crossing over will occur in meiosis. Then the product of mitosis is the two diploid daughter cells, whereas the product of meiosis is the four haploid daughter cells. 4 cells will be formed and the chromosomal status will be haploid. Then the daughter cells are completely identical to the parent cell, whereas the daughter cells will be genetically different from the parent cell. So thank you everyone. This is how we have completed the chapter. Thank you.